3. Paper O3 Biodiversity and Conservation. Module 39. Endemic Species of India Learning Objectives. Types of endemism. Habitat for endemics. Theories of endemism. Endemism in India. Endemic species of India. Introduction. The concept of endemism is central to the study of biogeography. The term endemism was coined by A.P. de Candolle 1855 for the distribution of an organism, plant, animal or microorganism in a limited geographical area. In ecological terms, it states that a plant or an animal lives only in a particular location, such as a specific island, habitat type, nation or other defined zone. It is the association of a biological taxon with a unique and well-defined geographic area. The cosmopolitan distribution or cosmopolitan is the antithesis of endemic, and refers to a taxon which is extremely widespread in many world regions. For example, highest numbers of endemic taxa are found in Australian region. All important islands and mountain chains except isolated piece of the country like Italy, from 48 to southwards possess endemics. Maximum proportion of endemism is found in West Australia and South African regions. All the southern land masses have great number of species confined to themselves, i.e. endemism is higher in old land masses than in young e.g., land of northern hemisphere, which are covered by the Pleistocene ice sheets, have lower number of endemics. Types of endemism. Two general classes of endemism are widely recognized, paleoendemism and neoendemism. A paleoendemic species. Paleoendemics means ancient endemics. These are the organisms, animals or plants that are restricted to an area because they have died out. 4. Paper O3 Biodiversity and Conservation. Elsewhere. A fossil record elsewhere is the best evidence for paleoendemism, e.g., in sequoia, Sequoiadendron, Lionothamnus. These are also described as those species which used to live in a large area in the past, but now live only in a smaller area. These species are often systematically isolated taxa, whose distribution areas represent the remnants of originally larger distribution ranges that have been reduced due to environmental changes. For example, species A is widely distributed throughout the whole mountain range. Any change occurring in the environmental conditions of the region leads to a reduction of the distribution of species A. Species A does not extinct entirely, it survives in a small area at the periphery of its former distribution range. Now, species A is a known as paleoendemic species of that area. These species are often systematically isolated taxa, whose distribution areas represent the remnants of originally larger distribution ranges that have been reduced due to environmental changes. For example, species A is widely distributed throughout the whole mountain range. Any change occurring in the environmental conditions of the region leads to a reduction of the distribution of species A. Species A does not extinct entirely, it survives in a small area at the periphery of its former distribution range. Now, species A is a known as paleoendemic species of that area. B. Neoendemic species. Neoendemism means that a species has recently appeared which is closely related to the main species or one that has formed following hybridization and is now classified as a separate species. This is a common process in plants, especially those which exhibit polyploidy. These species result from the divergent adaptation to differing environmental conditions, thereby leading to the formation of new species that are locally distributed. The evolution of neoendemic plant species is often triggered by polyploids. The endemic species may have a higher ploidy level than its related taxa, apoendemics, or it may be diploid while its related taxa have a higher ploidy level, petroendemics. In cases where the endemic taxon and its related taxa are of equal ploidy level, the endemic species are called schizoendemics. Apo, patro, and schizoendemics are further subdivisions of neoendemic. For example, species B immigrates to an area and colonizes the upper region of mountain chain. As a result, the population of species B gets separated and the two subpopulations are isolated from each other. Since the environmental conditions of the two subpopulations are not identical, they show different adaptations. The divergent evolution in the subpopulation. 5. Paper O3 Biodiversity and Conservation. May lead to the formation of new species or subspecies that are locally distributed and are called as neoendemic species. Habitat for endemics. The biological organisms have different patterns of distribution where they live on the globe. The territory where a species lives is called its distribution. It describes that where in the world that species naturally occurs. Therefore, based on their habitat of distribution on entire globe, species can be cosmopolitan, endemic or disjunct. Figure 1. For example, the distribution of polar bears restricted to Arctic region and lemurs occur naturally only in the island of Madagascar region is considered as the endemic species of that particular region. Another example of endemic species is bay checkerspot butterfly, i.e. Euphydryas edithobiensis, occurring in only one region in the San Francisco Bay Area. In contrast to this, brown rats, i.e. Rattus norvegicus, occurring everywhere, are considered as cosmopolitan species. Characters of endemism. They are localized in distribution because of their narrow ecological amplitude and are unable to invade in fresh areas. They lack potentially to migrate because of saturate genomes. Real endemics never migrate while neoendemics have the potential to migrate. The dispersal propagules are not able to sustain during migration to other area. It may be due to physical barriers. Theories of endemism. There are two main theories of endemism. The first theory believes that the last survivors of once flourishing flora which is now declining are the relics or epibiotics which are endemics. 6. Paper O3 Biodiversity and Conservation. However, second theory believes that these are recent and youthful forms in course of gradual extinction. According to Willis age and area hypothesis, most endemic species are considered to be youthful, i.e. youngsters, rather old relic. The concept of endemism includes two types of figure 1. Based on species distributional range of species, they can be categorized into various types. Organisms whose areas are confined to a single regions, endemics which are relatively youthful species, and epibiotics which are relatively old relic species. The theory is also known as age and area hypothesis. The first theory is supported by geographers, e.g., Sequoia semipenirans of the Central Valley of California and Oregon and S. gigantea of Sierra Nevada, which are endemic to their respective native homes, were extensively distributed in Cretaceous. Species distribution. Cosmopolitan. Widely distributed species example, brown rat. Endemic. Occurring in one geographic location example, polar bear. Disjunct. Occurring in two or more regions and that are separated from each other example, Nothofagus. 7. Paper 03 Biodiversity and Conservation. And tertiary periods. The supporters of second theory have the examples of primula, impatiens and rhododendron, etc. According to this theory, area is directly proportional to its age in the scale of evolution. So, a small area of distribution shows relatively young in age, e.g., Coleus is distributed on the summit of the dry Rittigala Mountains in Sri Lanka, with two species C. elongatus and C. barbatus. C. elongatus is endemic and C. barbatus is widely distributed in tropical Asia and Africa. Willis believed C. elongatus to be derived from C. barbatus. Factors responsible for endemism. Factors responsible for the production of endemics are natural crossing among the closely related plants growing under favorable conditions and mutations. If the condition of isolation is developed, the effect becomes more pronounced. Endemism is found in isolated, e.g., islands, isolated areas, etc. Mountains also have more endemic species as they are isolated, e.g., 70% sp. of Himalayas is endemic. Climate also is one of the factors, e.g., north of Himalaya is dry plateau of Tibet and South Himalayan range has alluvial fertile soil. According to Chatterjee, the percentage of endemic species of dicot plants in India is more than 50. Maximum endemic plants are found in the Himalayas and South India. Indo-Gangetic plains have a very small number of endemic species. Stebbins, 1942, has given a genetic explanation for the endemic. He told that such taxa have depleted their store of genetic variation, biotype depletion, and they are unable to expand their range. There are multiple causes of rarity and endemism. Three primary factors describe the distribution of endemics. Geographical area. Ecological role of species isolation. 8. Paper 03 Biodiversity and Conservation. Stebbins, 1980, has given the gene pool niche interaction theory to explain origin of rarity and endemism. According to theory, the primary cause of localized or endemic distribution patterns is adaptation to a combination of ecological factors that are themselves localized. Factors of soil texture and chemical composition are the most important but by no means the only ones. Next to the climatic and adaptive factors, those inherent into the gene pool of the population are of critical importance. They include the total amount of variability, the amount of variability that can be released at any one time, and the amount of variation that can be generated with respect to those particular characteristics that affect most strongly the establishment of new population. Endemism in India. India is a tropical country that is one of the mega-diversity centers, with only 2.4% of the world's land area, accounts for 7-8% of all recorded species, including over 45,000 species of plants and 91,000 species of animals. It is situated at the tri-junction of the Afrotropical, Indo-Malayan and Palearctic realms, all of which support rich biodiversity. Being one of the 17 identified megadiverse countries, India has 10 biogeographic zones and is home to 8.58% of the mammalian species documented so far, with the corresponding figures for avian species being 13.66%, for reptiles 7.91%, for amphibians 4.66%, for fishes 11.72% and for plants 11.80%. 4 of the 34 globally identified biodiversity hotspots, namely the Himalaya, Indo-Burma, the Western Ghats, Sri Lanka and Sundaland, are represented in India. The diverse physical features and climatic conditions have resulted in a variety of ecosystems such as forests, grasslands, wetlands, and desert, coastal and marine ecosystems which harbor and sustain high biodiversity and contribute to human well-being. In India, there are following 4 mega-endemic centers. 1. Indo-Burma covering Mizoram, Manipur, Nagaland, Meghalaya, Tripura and Andaman Islands. 9. Paper 03 Biodiversity and Conservation. 2. Himalaya covering Jammu and Kashmir, Himachal Pradesh, Uttarakhand, northern part of West Bengal, Darjeeling, Sikkim, northern part of Assam and Arunachal Pradesh. 3. Western Ghats falls within the states of Tamil Nadu, Kerala, Karnataka, Goa, Maharashtra and Gujarat. 4. The Sundaland covering the Nicobar Islands. Peninsular India is the richest endemic center. It harbors nearly 82% of the total endemic genera of the country. Peninsular India has a high concentration of endemic genera, 40 genera. There is no endemic family in India. There are only 49 genera endemic to India, of which 36 are unspecific. Hilltops provide a good environment for endemism next to islands. The species richness and high endemism in the Western Ghats is due to varied latitudinal and altitudinal gradients with varied rainfall and temperature. India has large number of endemic species. Endemic. The term, endemic, is used to denote a species, genus or other group confined to a small area like single island, a group of islands, a mountain chain or a comparatively small country like South Africa or West Australia, largely bounded by sea or by a marked alteration of climate. Endemic species (SN), endemic species, is one that is only found in a particular region and nowhere else in the world. Since these species are not widespread and may be confined to only 1 or 2 protected areas, they are of great conservation concern. A plant may be said to endemic to a certain state, to a country or to a continent. Although, there is no country or islands that have all its species endemic, yet it is very common to find genera with all their species endemic. Endemic species of India. The numbers of endemic plant under different plant groups mentioned in the 5th Report of Convention of Biological Diversity, CBD 2014 reported by Botanical Survey of India are given. 10. Paper 03 Biodiversity and Conservation. In Table 1. According to this report data, nearly about 4,045 species of flowering plants angiosperms endemic to India are distributed amongst 141 genera belonging to 47 families. These are concentrated in the floristically rich areas of northeast India, the Western Ghats, the northwest Himalaya and the Andaman and Nicobar Islands. Some examples of endemic plant species of India are Rhododendron sp. Ericaceae, Beaumontia grandiflora, Apocynaceae, Eleusine coricana, Poaceae, Caryota orens, Ericaceae, Agel marmelos, Rutaceae, Crotolaria uncia, Fabaceae, Ficus religiosa, Mauriceae, and Sesamum indicum petalaceae, and other plant species belong to families like Rubiaceae, 6 genera, Rosaceae, Astraceae, Primulaceae and Acanthaceae, etc. Some examples of the endemic animal species found in India are lion-tailed macaque, Nilgiri langur, brown palm civet and Nilgiri tar. Table 1. Three. Paper 03 Biodiversity and Conservation. Module 27. Endangered. 1. Endangered animal species of India. In previous modules we have learned about endemism. In this module we will study about endangered species of India. Endangered species. A plant, animal or microorganism that is in immediate risk of biological extinction is called endangered species or threatened species. In other words, these are those species whose life is under risk or threat and which are about to be extinct. These species which was native to a region and their population strength has reduced from 50% to 5%, such species are known as endangered species. Many factors are responsible for this which may be natural or man-made. Endangered plant species of India. The flora of India is one of the richest in the world due to the country's wide range of climate, topology, and environment. There are over 15,000 species of flowering plants in India which account for 6% of all plant species in the world. Many plant species are being destroyed, however, due to their prevalent removal. Roughly 1/4 of all plant species in the world are at. 4. Paper 03 Biodiversity and Conservation. Risk of being endangered or going extinct. The combination of global warming and habitat destruction is the sole reason for the disappearance of many plants. Though there are thousands of interesting and unusual plants, here are some common plants which have become rare and endangered species in the past 30 years due to habitat destruction. The combination of global warming and habitat destruction is the sole reason for the disappearance of many plants from Earth's face. Though there are thousands of interesting and unusual plants here are some common plants which we have seen 30 years back have become rare and endangered species. The examples of some endangered plant species were represented in the Table 1. Endangered animal species of India. India, a megadiverse country with only 2.4% of the world's land area, accounts for 7-8% of all recorded species, including over 45,000 species of plants and 91,000 species of animals. Being one of the 17 identified megadiverse countries, India has 10 biogeographic zones and is home to 8.58% of the mammalian species documented so far, with the corresponding figures for avian species being 13.66%, for reptiles 7.91%, for amphibians 4.66%, for fishes 11.72% and for plants 11.80%. 4 of the 34 globally identified biodiversity hotspots, namely the Himalaya, Indo-Burma, the Western Ghats, Sri Lanka and Sundaland, are represented in India. India, with only about 2.4% of world's total land surface, it is known to have over 6.7% of the animal species that the world holds. This percentage represents nearly 96,373 known species, including 63,423 insect species. In India, 450 plant species have been identified as endangered species. 100 mammals and 150 birds are estimated to be endangered. 5. Paper 03 Biodiversity and Conservation. Figure 1, representing the total numbers of endemic and threatened endemic species of India. Source, IUCN 2013. Reasons for species becoming endangered. In India, 450 plant species have been identified as endangered species. 100 mammals and 150 birds are estimated to be endangered. India's biodiversity is threatened primarily due to I. Habitat destruction. E. Degradation and E. Overexploitation of resources. Factors affecting endangered species. Human beings dispose wastes indiscriminately in nature, thereby polluting the air, land and water. These pollutants enter the food chain and accumulate in living creatures, resulting in death. Overexploitation of natural resources and poaching of wild animals also leads to their extinction. 6. Paper 03 Biodiversity and Conservation. Climate change brought about by accumulation of greenhouse gases in the atmosphere. Climate change threatens organisms and ecosystems and they cannot adjust to the changing environmental conditions, leading to their death and extinction. Table 1. List of some endangered plant species of India. Botanical name of plant species. Common name. State status. Abutilon indicum Indian mallow, Thuthi Tamil, and Athibala Sanskrit. Tamil Nadu, rare. Acacia planifrons umbrella tree, Kudable Tamil. Tamil Nadu, rare. Actinodaphne lawsoni Kerala, threatened. Amentotaxis asamica Assam, Katganyu Arunachal Pradesh. Threatened Belisynapsis viviparous spider wart, Madhya Pradesh, rare and endangered. Saropagia odorata Jimikanda, Gujarat, Melgat tiger, Rajasthan, and Salsette. Island, endangered. Chlorophytum malabaricum Malabar lily Tamil Nadu. Threatened Chlorophytum tuberosum muesli Tamil Nadu. Colchicum luteum Himachal Pradesh. Rare and threatened Diospyros salibica ebony tree Karnataka. Threatened Lotus corniculatus bird's foot Gujarat. Rare. Nymphaea tetragona Jammu. Endangered. Kashmir. Threatened Polygala irregularis milkwort Gujarat. Rare. 7. Paper O3 Biodiversity and Conservation. Silotum nudum moa, skeleton, fork fern, and whisk fern. Karnataka. Rare. Pterospermum reticulatum malayarum, Malavorum Kerala. Rare. Tamil Nadu. Threatened source. 62 and source equals LNMS and TBM equals ish and sa. Endangered animals of India. The world is filled with endangered species of animals, many of which can be found in India. While some of these species are critically endangered, some are near threatened and some are even extinct. There are some species that are left in such few numbers that your next generation may not be able to ever see them, if nothing is done for their conservation. While we hope that more efforts are taken to conserve these species, here's a list of some endangered animals that you can find in different parts of India. Table 2. List of various endangered species of animals of India. S number. Animal name description location. 1. Bengal tiger. The Bengal tiger is the national animal of both India and Bangladesh. Tadoba National Park, Ranthambore National Park, Sundarbans National Park, Sariska Tiger Reserve, Jim Corbett National Park, Bandhavgarh National Park. 8. Paper O3 Biodiversity and Conservation. 2. Asiatic lion Indian lion Persian lion. It differs from the African lion by less inflated auditory bully, a larger tail tuft and a less developed mane. Gir Forest National Park, Gujarat. 3. Snow leopard. The snow leopard is a large cat native to the mountain ranges in Central and South Asia. Snow leopards have long, thick fur, and their base color varies from smoky gray to yellowish tan, with whitish underparts. Hemis National Park, Ladakh, Nanda Devi National Park, Uttarakhand, Dibang Wildlife Sanctuary, Arunachal Pradesh, Khyber Wildlife Sanctuary, Lahaul Spiti, Himachal Pradesh, Pin Valley National Park, Lahaul Spiti, Himachal Pradesh, Great Himalayan National Park, Kulu, Himachal Pradesh. 4. Blackbuck. The blackbuck is an ungulated species of antelope and it is near threatened. The main threat to this species is poaching, predation, habitat destruction, overgrazing, inbreeding and sanctuary visitors. Gwindi National Park, Tamil Nadu, Rolapadu, Andhra Pradesh, Chilka, Orissa. 9. Paper O3 Biodiversity and Conservation. 5. The Nilgiri tar. The Nilgiri tar is an ungulate, endemic to the Nilgiri Hills. Nilgiri tars are stocky goats with short, coarse fur and a bristly mane. Araviculam National Park, Nilgiri Hills, Animalai Hills, Periyar National Park, Palni Hills. 6. Kashmir red stag Hongal. The Kashmir stag, also known as Hongal, is a critically endangered species. This deer has a light rump patch without including the tail. Each of its antlers consists of 5 tines. Dachigam National Park, Rajparian Wildlife Sanctuary, Avera Aru, Sindh Valley, and in the forests of Kishtwar and Baidarwa, all of which are a part of Jammu and Kashmir. 7. Lion-tailed macaque. The lion-tailed macaque is an Old World monkey, endemic to the western Ghats of South India. Its outstanding characteristic is the silver-white mane which surrounds the head from the cheeks down to chin. Silent Valley National Park, Kerala, Papanasam, part of the Kalakad Mundanthari Tiger Reserve, Tamil Nadu and Sirsi Honavara rainforests of northwestern Ghats in Karnataka. 8. Indian bison Gaur. Indian bison is the largest extant bovine, native to South Asia and Southeast Asia. The bisons are highly threatened by poaching for trade to supply international markets. Arinar Anna Zoological Park, Chennai and Chinnar Wildlife Sanctuary, Kerala. 10. Paper O3 Biodiversity and Conservation. 9. One-horned rhinoceros. The one-horned rhino is a large mammal, found in Assam. Excessive hunting has reduced the number of this species. These rhinos are killed to saw off their horn, which are sold at a very high value. Kaziranga National Park, Dudhwa Tiger Reserve, Kobatora Wildlife Sanctuary. 10. Indian bison Gaur. Indian bison is the largest extant bovine, native to South Asia and Southeast Asia. The bisons are highly threatened by poaching for trade to supply international markets. Arinar Anna Zoological Park, Chennai and Chinnar Wildlife Sanctuary, Kerala. International and national efforts to protect endangered species. International efforts. Convention on International Trade in Endangered Species, CITES, an international treaty to help protect endangered wildlife. This treaty is now signed by 160 countries. CITES lists 900 species that cannot be commercially traded as live specimens or wildlife products as they are in range of extinction. CITES restricts trade of 2,900 other species as they are endangered. National efforts. Various organizations deal with the protection of endangered species of India. These are Wildlife Protection Society of India (WPSI). It is a registered non-profit organization funded by a wide range of Indian and international donors. The Society's board. 11. Paper O3 Biodiversity and Conservation. Members include leading conversationalists and business people. It was founded in 1994 by Belinda Wright, its executive director, who was an award-winning wildlife photographer and filmmaker till she took up the cause of conservation. WPSI main aim has been to bring a new focus to the daunting task of tackling India's growing wildlife crisis. It does this by providing support and information to government authorities to combat poaching and the escalating illegal wildlife trade, particularly in wild tigers. B. Wildlife Trust of India (WTI), non-profit organization set up to prevent destruction of India's wildlife, especially endangered species tigers, elephants, rhinos, and threatened habitats, aids research and projects, educates public, acquires land for wildlife conservation. C. Wildlife Conservation Society. Indian program of the Global Wildlife Society that focuses on endangered megafauna in protected reserves, focuses on studying tigers, leopards, and their ungulate and primate prey species. D. Wildlife Institute of India. It was established in Dehradun, Uttarakhand, India in 1982 with an aim to provide training and courses, and promote wildlife research and management. Is actively involved in research of biodiversity and major wildlife issues in India. E. Wildlife Conservation Trust WCT. It works to protect the vulnerable animals and biodiversity of India through spreading awareness across forest departments and NGOs. WCT even trains individuals for wildlife conservation. F. Conservation India. C. It is a non-profit, non-commercial portal which is working in order to promote strategies for wildlife conservation. C. Believes in working on ground rather than simply speaking about wildlife conservation. G. People for Ethical Treatment of Animals. PETA. India has been functional in Mumbai since 2000. It works to survey the segments of society where animals are poorly treated, like in food or leather industries, entertainment industry, etc. PETA. India is known for spreading public awareness, triggering animal rescue missions and 12. Paper 03 Biodiversity and Conservation. Legislative work, but most importantly for improving and saving the lives of innocent animals. H. Nature Conservation Foundation NCF works in varied animal habitats, including that of coral reefs, tropical rainforests and as far as the Himalayas. NCF works to know the fundamental needs of elephants, snow leopards and even spiders and corals. NCF even conducts research in order to address the impact of commercial usage and its effects on ecology. I. Center for Wildlife Studies CWS. It was established in 1984 in Bengaluru with an aim to promote conservation of the tiger and other large mammals, along with equal emphasis for ecology through collaboration with state and central governments. Today, CWS is an internationally known institute for excellence in the field of wildlife research and conservation. J. The Corbett Foundation. The Corbett Foundation involves actions taken by passionate men and women who wish to protect wildlife and bring forth harmony. This foundation raises funds, conserves various ecosystems, draws the government's attention towards major wildlife issues and promotes wildlife research projects. This foundation works for protection of other regions such as Bandhavgarh, Kutch and Kanha as well. K. Bombay Natural History Society. Bombay Natural History Society BNHS, established in 1883, is associated with wildlife research and conservation in India for over 130 years. BNHS mainly focuses on research, education and public awareness about wildlife and nature. National Conservation Plan. For the conservation of various endangered species, several projects has been initiated. The list of various is given below. Vulture conservation. Because of a serious decline in the population of the main species of vulture, a series of meetings and seminars are being held in India in order to 13. Paper 03 Biodiversity and Conservation. Address the need for vulture conservation. The national workshop held in New Delhi in 2004, for the same cause was attended by Chief Wildlife Wardens, BNHS Bombay Natural History Society, Sekhan Salim Ali Center for Ornithology and Natural History, Coimbatore Kir Gujarat Ecological and Education Research Foundation, Gandhinagar Department of Animal Husbandry. B. GOIUNDP Sea Turtle Project. Executed by Ministry of Environment along with Wildlife Institute of India, Dehradun, the Government of India. Sea Turtle Project works to protect sea turtles. Launched in 1999, this project carries out rigorous surveys along the coastal areas, checks the community and legal conservation, creates awareness, gathers information about main nesting areas and also provides training to fisheries managers, researchers and biologists. C. Crocodile Conservation Project. It was launched in 1976. The main objective of this project is to protect the three most endangered species of crocodile, the freshwater crocodile, gharials and saltwater crocodiles. The Crocodile Conservation Project undertakes captive breeding of various species of crocodiles, looks after the hatching and rearing of gharials in order to decrease the mortality rate of crocodiles. Under Gharial Rehabilitation Project, about 200 gharials were sheltered and consequently released in the Ramganga River in Corbett National Park. D. Project Elephant. It was initiated by the Government of India in 1992 as a centrally sponsored scheme. The main objective of Project Elephant is to conserve elephants, their habitat, work for issues such as human-elephant conflict and look after the welfare of domestic elephants. E. Project Tiger. Project Tiger is a scheme centrally sponsored by the Ministry of Environment, Forest and Climate Change. This scheme covers around 47 tiger reserves located across more than 17 regions populated with tigers. Project Tiger mainly focuses on conducting assessments of number of tigers, their habitat, hunting habits, etc. All these activities are sanctioned by the Tiger Task Force. 14. Paper 03 Biodiversity and Conservation. F. Indian Rhino Vision 2020. India Rhino Vision (IRV) 2020 is an effort of pact made by the Bodoland Territorial Council, WWF World Wildlife Fund IRF International Rhino Fund and the U.S. Fish and Wildlife Service. The main objective of IRV is to conserve at least 3,000 greater one-horned rhinos in Assam, India by 2020. Laws and Acts. In India, various laws were made to protect plants and animal species. These were followings: I. Indian Forest Act, 1927. E. Wildlife Protection Act, 1972. E. Forest Conservation Act, 1981. IV. Environment Protection Act, 1986. B. The Biological Diversity Act, 2002. A detailed account of these acts is given in Paper 13, Environmental Laws and Policies References. Conservation International. IUCN 2013 IUCN Red List of Threatened Species. IUCN Species Survival Commission, Gland, Switzerland. 15. Paper 03 Biodiversity and Conservation. Paper coordinator. Professor Dave. Module 28. International Union for Conservation of Nature IUCN Categories of Threatened Species. Learning outcomes. In this module we will learn about International Union for Conservation of Nature IUCN Categories of Threatened Species. Introduction. The International Union for Conservation of Nature IUCN is an international organization working in the field of nature conservation and sustainable use of natural resources. It is the leading international environmental organization. Its headquarters is at Gland, Switzerland. History. The IUCN was founded on the 18th of October 1948 at Fontainebleau, France. It is the world's most comprehensive inventory of the global conservation status of species. The organization is funded by governments, member organizations, bilateral and multilateral agencies, etc. It is the world's main authority on the conservation status of species. It is headquartered in Gland, Switzerland. India became a state member of IUCN in 1969. The office of IUCN in India is located in New Delhi, established in 2007. Logo of IUCN. IUCN has produced several key international environmental agreements, namely, A, the Convention on Biological Diversity, CBD. B, the Convention on International Trade in Endangered Species, CITES. C, the World Heritage Convention, and Paper 03 Biodiversity and Conservation. D, the Ramsar Convention on Wetlands. Functions of IUCN. The organization is best known for compiling and publishing the IUCN Red List of Threatened Species, which assesses the conservation status of species worldwide. The mission of IUCN is to persuade, promote and help societies to preserve the diversity of nature. Its aim is to protect nature and promotes its sustainable utilization of natural resources. It also integrates matter of poverty alleviation, climate change, and biodiversity and gender equality. Activities of IUCN. IUCN works over a wide range of themes related to conservation, environmental and ecological issues. Some of them are given below. Source: IUCN. A business and biodiversity aims to transform business values, manages and invests in nature, highlighting the opportunities and benefits of a more sustainable approach. B. Climate change. Assessing the risks of climate change, practical nature-based solutions centered on better conservation, management and restoration of natural ecosystems. C. Ecosystem management. IUCN works to have healthy ecosystems to provide goods, food and water, and services, climate regulation and protection from natural hazards. D. Forest conservation. E. Protection of marine and polar environments. F. IUCN Global Species Program is to save species for people and nature. G. Water conservation and management. H. Natural World Heritage Sites. I. IUCN Global Protected Areas Program. In addition to this, several other activities are undertaken by IUCN. The details of the latest activities of IUCN can be obtained from its official website. IUCN has generated several resources, such as A. Conservation tools. B. IUCN Red List of Threatened Species. C. IUCN Red List of Ecosystems. D. Key Biodiversity Areas. Paper 03 Biodiversity and Conservation. The details of some of them are given below. Conservation tools. The key objective of IUCN is to share the knowledge. IUCN's knowledge products consist of conservation databases and tools. The list of conservation databases developed by IUCN is given below. The IUCN Red List of Threatened Species. The IUCN Red List of Ecosystems assesses the World Database on Key Biodiversity Areas. Protected Planet assesses ECOLEX, a gateway to environmental law. A. IUCN Red List of Threatened Species. The IUCN Red List of Threatened Species was created in 1964. This is the world's most inclusive record of worldwide conservation status of biological species. It is one of the most well-known objective assessment systems for classifying the status of plants, animals, and other organisms threatened with extinction. The International Union for Conservation of Nature (IUCN) unveiled this assessment system in 1994. It contains explicit criteria and categories to classify the conservation status of individual species on the basis of their probability of extinction. The species are categorized into 9 groups by IUCN Red List as shown in the Figure 1. Paper 03 Biodiversity and Conservation. Paper 03 Biodiversity and Conservation. Logo of IUCN Red List of Threatened Species. Criteria for classification. The IUCN system uses a set of 5 quantitative criteria to assess the threat or extinction risk of a given species. These are the followings. 1. The rate of population decline of a given species. 2. The geographic range. 3. Population size. 4. Species size or its habitat area. 5. Whether these above quantitative properties indicate a high probability of extinction of species in the wild. Figure 1. Showing IUCN Red List various categories. Table 1. Representing various categories under Red List. All species. Extinct in the wild. Critically. Endangered vulnerable nor threatened. Least concern. Data. Deficient. Not. Evaluated. IUCN category description. Extinct X, a taxon is E when there is no reasonable doubt that the last individual has died. For example, dodo extinct in the wild. EU, a taxon is EU when it is known only to survive in cultivation, in captivity or as a naturalized population, or populations well outside the past range. For example, Alagoas curassow. Critically endangered CR, a taxon is CR when it is considered to be facing an extremely high risk of extinction in the wild in immediate future. For example, gharial. Endangered N, a taxon is N when the best available evidence indicates that it is facing a risk of extinction and has become less in number. For example, tiger. Vulnerable. Boo. A taxon is vulnerable when it is not CR or EN but it facing a high risk of extinction in the wild in the medium-term future. For example, polar bear. Near threatened. NT. A taxon is NT when it has been evaluated against the criteria but does not qualify for CR, N or BU now, but is close to qualifying for or is likely to qualify for a threatened category in the near future. For example, blue-billed duck. Least concern. LC. A taxon is LC when it has been evaluated against the criteria and does not qualify for CR, N, BU or NT. Widespread and abundant taxa are included in this category. Data deficient. DD. A taxon is DD when there is inadequate information to make a direct or indirect assessment of its risk of extinction based on its distribution and or population status. A taxon in this category may be well studied and its biology well known, but appropriate data on abundance and or distribution are lacking. Data deficient is therefore not a category of threat. Not evaluated. Nay. A taxon is nay when it has not yet been evaluated against the criteria. Paper 03 Biodiversity and Conservation. B. IUCN Red List of Ecosystems. The IUCN Red List of Ecosystems is a global standard to access the conservation status of ecosystems. It is applicable at local, national, regional and global levels. It is based on a set of rules, or criteria, for performing evidence-based, scientific assessments of the risk of ecosystem collapse, as measured by reductions in geographical distribution or degradation of the key processes and components of ecosystems. The IUCN Red List of Ecosystems is working towards 5 major targets. 1. Classify and list the world's ecosystems and document their status. 2. Focus not only on threatened ecosystems but also on those that are in good condition as a result of active management, and so highlight best practices in ecosystem management. 3. Establish a secretariat to manage the Red List of Ecosystems process in collaboration with the IUCN Red List of Threatened Species. 4. Enhance technical and institutional capacity for ecosystem red listing at national, regional and global levels. 5. Develop strong linkages between good ecosystem management and sectors which are not necessarily focused on conservation, e.g. national and economic planning, livelihood improvement, and the private sector. The details regarding development of the Red List of Ecosystems and risk assessment criteria can be obtained from the website of IUCN. IUCN 2017-2020 Program and Future Prospects. The IUCN Programs Working for People and Nature provides the framework for planning, implementing, monitoring and evaluating the conservation work undertaken by the commissions and the Secretariat with and on behalf of IUCN members. At present, IUCN Program 2017-2020 was approved by member organizations at IUCN's World Conservation Congress Figure 2. The IUCN Program 2017-2020 has 3 main priority areas: of valuing and conserving nature. B. Promoting and supporting effective and equitable governance of natural resources. C. Deploying nature-based solutions to societal challenges. The aim of IUCN is that up to 2025, to assess the global conservation status of all of the world's terrestrial, freshwater, marine and subterranean ecosystems and to create the first IUCN Red List of Ecosystems of the world. Paper 03 Biodiversity and Conservation. Paper 03 Biodiversity and Conservation. Figure 2. Proposed timeline for development of the IUCN Red List of Ecosystems to achieve global coverage. References. Red List of Threatened Species, as an indicator of biodiversity trend. IUCN 2013 IUCN Red List of Threatened Species. IUCN Species Survival Commission, Gland, Switzerland. 2008. IUCN process begins at Barcelona WCC. 2012-2014. IUCN Red List of Continental Ecosystems of the America. 2015. Standardized Protocols and Online Tool Full available 2015-2020, expansion to other biological realms and regions initiated. 2012. Synthesis and presentation at Jeju WCC. 2009. IUCN Red List established 2010. Draft Red List categories and criteria. 2011. Global consultation and testing initiated. 2020-2025. First global IUCN Red List of Ecosystems completed. Cause of biodiversity loss. 2. Understand the urgent need of biodiversity conservation. 3. Different modes of biodiversity conservation. 4. Biotechnological approaches available for conservation. 2. Concept map. 3. Description. 3.1 Biodiversity conservation. Biodiversity exists in 3 different levels: genes, species, and ecosystems in a given geographical area. Each of the components has its own composition, structure, and function in environment. In recent past, ever-increasing loss of biodiversity has posed a serious threat to the survival of fauna and flora present on this planet. Destruction of the habitat of plants and animals, introduction of exotic species, overexploitation of animals and plants, overpopulation of human and overconsumption of natural resources are the root causes of biodiversity destruction. There is an urgent need for the conservation of biodiversity. Biotechnology offers new opportunity of conserving biodiversity. Biotechnology including tissue culture, micropropagation, marker-assisted breeding, conventional breeding, transgenic crops, and genomics. Figure 1. Systematic representation of different modes of biodiversity conservation. Biotechnological approaches: gene bank, tissue culture and cryopreservation. Botanical gardens. Zoological parks. Aquaria and home gardens are all quite beneficial for conserving biodiversity in many ways. Biotechnology has been used to improve and enhance crop productivity, as well as to conserve and utilize the various aspects of biodiversity. The global concern of biodiversity preservation initiated either by in situ or ex situ methods. In situ methods protect fauna, flora and their natural habitat. On the other hand, ex situ methods involves preservation and maintenance of biodiversity outside their natural habitat. Classical methods of plant propagations have certain limitations in terms of rapid production of plant propagules and their long-term conservation. So, the biotechnological methods such as plant cell culture, plant tissue culture, embryo culture, anther culture, etc. are quite applicable and useful techniques for ex situ conservation. Nowadays, loss of specific species, extinction, or decreases in number of particular organisms, endangerment, are taking place in different parts of the world at a rapid speed. Domestic and wild animal species are undergoing a profound erosion process. Conservation of biodiversity through establishment of protected areas like national park, wildlife sanctuary, biosphere reserves, marine reserves, etc. are very effective in controlling the loss of biodiversity. Although biotechnology is commonly thought of as recombinant DNA technology, it is used here in a broader term to include tissue culture, cryopreservation, plant micropropagation and animal regeneration from early embryos. Biotechnology influences germplasm conservation in several ways. It provides alternatives in some cases to conserving whole organisms, it can assist with the exchange of germplasm, and the techniques of molecular biology can be applied to the problems of managing and using germplasm and influence results from the increased demand for germplasm and conservation services by the biotechnologists themselves. In situ conservation focuses on preserving the genetic variation in the location it has been encountered originally, i.e. in its natural habitats either in the wild or in traditional farming system. In situ conservation can be carried out at any level, in any country, without the need for special skills or technology. Properly planned, it can incorporate farmer and industry breeding as well as continued use of the resources. On the other hand, ex situ involves conservation outside the native habitat and is generally used to safeguard populations in danger of destruction, replacement or deterioration. Approaches to ex situ conservation include methods like seed storage, field gene banks and botanical gardens. Ex situ conservation of animal genetic resources includes cryogenic preservation techniques, the collection and freezing in liquid nitrogen of semen, ova or embryos, or the preservation of DNA segments in frozen blood or other tissues. It also encompasses the captive breeding of wild or domesticated species in zoos or other locations away from their natural environment. Ex situ gene banks have similar advantages and disadvantages to those used to hold plant germplasm. In situ conservation enables animal populations to continue to evolve and be selected for use in their natural environments. It is particularly important for species or within geographical regions where cryogenic preservation is not well developed or available. Biotechnological approaches of biodiversity conservation come under ex situ mode of conservation. 3.2 Various biotechnological approaches in biodiversity conservation. The main objective of germplasm conservation is to maintain constant preservation of germplasm as it can be available at any time. Biotechnological approaches are imperative for rapid multiplication and conservation of the critical genotype of medicinal plants. These include in vitro propagation through tissue culture, genetic transformation, cryopreservation, DNA banks, etc. 3.3 Tissue culture techniques. Plant tissue culture (PTC). PTC is a quick, independent and efficient in vitro technique to propagate plants under sterile microenvironmental conditions. It is a very effective method of cloning of plant material and to develop disease-free clean plant stock for propagation. Any plant cell has the power of cellular totipotency to be differentiated into whole plant in the process of plant tissue culture. The main objective of PTC is the enhancement of plant production rate by quick regeneration of plants in the absence of seed or otherwise by using the seeds which have very low chances of germination. Tissue culture techniques can be applied in germplasm conservation of medicinally important plants. Those are of important source of pharmaceutical compounds. It can be applied for regenerating different clean disease-free stock of plants in the field of agriculture, horticulture, floriculture and pharmaceutical industry to enhance production. Rapid and mass propagation of plant species and their long-term germplasm storage can be achieved in a small space within short time period, with no damage to the existing population using PTC methods. Microorganism removal is a critical factor that must be strictly controlled during in vitro collecting of plant material. Bacteria and fungi develop rapidly as saprophytes in culture media, and since their nutritional requirements are basically the same as the explants, they compete with the explant for nutrients and they can produce phytotoxic metabolites that affect plant growth. Tissue culture techniques are of great interest for collecting, multiplication and storage of plant germplasm and are very useful for conserving plant biodiversity, including a genetic resources of recalcitrant seed and vegetatively propagated species, B, rare and endangered plant species, and C, biotechnology products, such as elite genotypes and genetically engineered material. Tissue culture systems allow propagating plant material with high multiplication rates in an aseptic environment. Figure 2. Steps in plant tissue culture. Source: Pinterest. Micropropagation 3.4 Micropropagation. Micropropagation help in the production of virus-free plants and is successful for those plant species which have difficulties in propagation using conventional methods. It is an in situ conservation of plant species having special phenotypic characters by introduction of regenerated plantlets directly into its natural habitat. In a short period of time and limited space, it allows the production of numerous plant species. The selection of the explants is very important for the successful micropropagation which decides the final fate of the developed plantlets via callus or without formation of calluses. The micropropagation of Lavandula pedunculata was done for essential oil production without affecting natural resources. The micropropagation is rapid and has been adopted for commercialization of important plants such as banana, apple, pears, strawberry, cardamom, many ornamentals. 3.5 Somatic embryogenesis and organogenesis. The development of somatic embryo from a single or multicells is known as somatic embryogenesis. Plant genetic transformation and gene cloning depend on the plant tissue culture practices for their regeneration on suitable media. Plant propagation through somatic embryogenesis helps to obtain a large number of plants irrespective of seasons and provides several advantages over traditional methods. The main advantage of SE is the production of numerous plantlets from a single cell which provides an option for their screening and evaluation. Biotechnological tools have an important contribution in the production of homozygous line from gametic embryogenesis. The de novo organ synthesis such as buds, roots and shoots from cultured tissues is called organogenesis. Plant mature cells have the capability to reverse their state of differentiation and produce new organs under cultured conditions. In vitro organogenesis, the differentiation and redifferentiation phases are commonly characterized. The balance of exogenous auxin and cytokinin in the medium is essential for de novo organogenesis. Thus, organogenesis entails the regulation of cell division, cell expansion, cell and tissue type differentiations, and patterning of the organ as a whole. 3.6 Animal tissue culture. The two principal methods for conserving animal germplasm in vitro are as frozen semen and embryos. Both methods require the maintenance of female animals for either insemination or implantation. Embryos have an advantage over semen because they provide the complete genotype. In some domestic species, embryos for transfer or cryopreservation are collected from animals that have received hormonal treatment to induce an excess of eggs for insemination. The resulting embryos are evaluated for their potential to produce a pregnancy or to withstand freezing. Embryos from cattle, sheep, goats, and horses have all been frozen in liquid nitrogen, thawed, and successfully implanted. 3.7 Cryopreservation. Cryopreservation is one of the biotechnological method of ex situ plant conservation and applicable for long-term storage of plant genetic material. Cryopreservation is extremely helpful method to conserve rare, endangered, threatened species. There are two types of cryopreservation protocols that basically differ in their physical mechanisms: classical cryopreservation procedures, in which cooling is performed in the presence of ice, and the procedures based on vitrification, in which cooling normally takes place without ice formation. Classical freezing method includes the successive steps such as pre-growth of samples, cryoprotection, slow cooling 0.52 C per minute to a determined pre-freezing temperature usually around -40 C, rapid immersion of samples in liquid nitrogen LN storage, rapid thawing and recovery. It needs technical skill as it has several steps such as freezing, storage, thawing, reculture of living plant cells during cryopreservation against cryogenic injuries. Maintenance of cryogenic cultures in lane at -196 degrees Celsius or in the vapor phase of lane at -135 degrees Celsius is in such a way that the viability of stored tissues is retained following rewarming. Certain compounds like TIMSO, dimethyl sulfoxide, glycerol, ethylene, propylene, sucrose, mannose, glucose, proline, acetamide etc. are added during the cryopreservation. These are called cryoprotectants and prevent the damage caused to cells by freezing or thawing by reducing the freezing point and supercooling point of water. Various cryopreservation methods are being used for various plant species such as vitrification, encapsulation dehydration and encapsulation vitrification. Cryopreservation using the encapsulation dehydration procedure has been very effective for freezing apices of different plant species from temperate and tropical origin. Vitrification comprises treatment of samples with cryoprotective substances followed by dehydration with highly concentrated plant vitrification solutions (PVS), rapid cooling and rewarming, removal of cryoprotectants and recovery. This procedure has been developed for apices, cell suspensions and somatic embryos of different species. Cryopreservation of seeds is a very valuable strategy for the long-term conservation of tropical and subtropical forest species biodiversity, as it avoids problems related to embryo isolation and in vitro handling. The cryopreserved tissue has considered as safer, clean, disease-free genetic stock. Virus-free strawberry plants could be preserved at 1000 C for about 6 years. Several grape plants have been stored for over 15 years by using a cold storage at temperature around 90 C and transferring them in the fresh medium every year. 3.8 DNA banks. DNA banking is also a potential method for the conservation of biological information by preserving the genomic DNA at low temperatures. The DNA isolation is easy and can be used extensively for the characterization and utilization of biodiversity. The implementation of such biotechnological tools on rare and endangered plant species may help in revival of their previous genes and their products which have been disappeared or inactivated in natural habitat. In gene banks, the plant and animal materials are conserved and are available for breeding, reintroduction, research and other purposes. DNA banks maintain a library of the given DNA sample and provide vital information to the conservation scientists. DNA samples may be of 3 kinds: I total genomic DNA, E DNA libraries, E individual clone DNA fragments. The maximum possible genetic diversity of the particular genetic stock can be maintained using in vitro gene bank technique. Some important DNA banks includes the Royal Botanic Garden, Kew, UK, presently the world's largest DNA bank, consisting of over 20,000 DNA specimens representative of all plant families. The U.S. Missouri Botanical Garden has collection of more than 20,000 plant tissue samples, and provide raw material for the extraction of DNA for its subsequent use in conservation methods. The International Plant Genetic Resources Institute (IPGRI), as well as the Consultative Group on International Agricultural Research (CGIAR), an International Center for Agricultural Research in the Dry Areas (ICARDA), is heavily involved in the conservation of rare and endangered plant species by maintaining in vitro gene bank. Animal gene banks development is limited by high costs of creation. The recovery and freezing of viable sperm recovered from the epididyms of slaughtered or castrated animals can be a cheap alternative for preserving male gametes. Nowadays, application of post-mortem sperm recovery from the epididymis is available for conservation of endangered species and in constructing semen banks. Preservation of epididymal sperm from accidental dead or slaughtered animals has been reported in several domestic and wild species as boar, cattle, goat, ibex, red deer and sheep. However, epididymal sperm extraction sometimes is not easily practicable in marginal areas, due to the lack of facilities and expertise near the farming areas of local breeds. 3.9 Molecular marker technology. 3.10 Biochemical marker. Different variants of the same enzymes having identical or similar functions are known as isozymes. They are powerful tools for the study of genetic variability within and between populations of plants and animals. They are useful when several taxa, accessions and individuals are to be compared, to identify clones and to examine the clonal structure of various plant populations. 3.11 Phytochemical markers. The discovery of phytochemicals from wild plant species is an achievement toward the eradication of the human diseases. With the advancement of modern techniques such as mass spectrometry (MS) and nuclear magnetic resonance spectrometry (NMR) combined with different separation techniques facilitated the identification and structural elucidation of these phytochemicals. These phytochemical analyses are valuable tools for taxonomic differentiation within species or for evaluating the effect of environmental factors. Variation in biosynthesis of these metabolites could be a result from both genetic and environmental factors, which play important roles in the development of phenotypic variations in plants. 3.12 DNA marker-based techniques. DNA-based markers either PCR-based or non-PCR-based. DNA markers assess genetic diversity based on polymorphism. The polymorphism may be defined as simultaneous occurrence of multiple phenotypic forms of a trait attributable conservation of rare and endangered plant species 3 to the alleles of a single gene or the homologue of a single chromosome within or between populations. Potential DNA-based markers are single-strand conformation polymorphism (SSCP), restriction fragment length polymorphism (RFLP), random amplified polymorphic DNA (RAPD), amplified fragment length polymorphism (AFLP), sequence characterized amplified regions (SCAR), simple sequence repeat (SSR), inter-simple sequence repeat (ISSR), single nucleotide polymorphism (SNP). Those are either PCR-based or non-PCR-based techniques. Molecular markers (SSCP). This technique is used for identification of point mutation and for polymorphism. The heterozygosity is easily detected as the mobility of ssDNA changes with change in its GC content. RFLP. The principle is based on the variation in the restriction site of the enzyme, which produces different restriction fragments on digestion. It is quite tedious and requires large amount of purified DNA. RAPD. RAPDs have been extensively used in assessing genetic relationship among various accessions of different plant species. RAPDs are very quick and easy to develop due to the arbitrary sequence of the primers. It is cost effective and easy to perform. AFLP. This technique is particularly useful for evaluating genetic diversity in those plant species where prior genetic information is not available. It is the modified form of the RFLP and uses PCR for the amplification of the restriction fragments generated on digestion of genomic DNA. More variation in the restriction site, more polymorphism would be generated. SCAR. The RAPD marker can be converted into SCAR. It is monolocous and more specific. These markers are reproducible and less sensitive to reaction conditions for amplification. These markers are based on sequencing of the unique band. The unique band is selected for the designing of longer primers that specifically binds to same loci of exact molecular weight is amplified in the PCR. SSR. These are tandemly repeated DNA sequences in the length. Variation in this marker is due to the DNA polymerase slippage during the replication of the SSR region. These markers show a high level of allelic diversity. The allelic polymorphism and codominant nature revealed by SSR markers have provided information on genetic structure and gene flow in natural plant populations. ISSR. ISSR marker is used for the evaluation of genetic diversity because the nucleotide repeats are distributed throughout the genome and has power which has been seen in the clonal plant species for the assessment of genetic diversity. ISSR's markers are quick, easy to handle and more reproducible than RAPD's markers. SNP. SNPs are used as molecular genetic markers in cultivar identification, genetic diversity studies and linkage mapping, characterization and phylogenetic studies, marker-assisted breeding, positional cloning and genetic comparative mapping. These markers occur in unlimited numbers. Figure 3. Systematic representation of different DNA-based molecular markers. of different DNA-based molecular markers. Environmental sciences. Paper O3 Biodiversity and Conservation. Biotechnological approaches to biodiversity conservation. 3.13 Gene transformation. Genetic transformation is a powerful tool for enhancing the productivity of novel secondary metabolites of limited yield. Genetic transformation facilitates the growth of medicinal plants with multiple durable resistances to pests and diseases. Likewise, transgenes or marker-assisted selection may assist in the development of insect, pest, and drought, salinity-resistant plants, which will be needed to fulfill the ever-increasing world's need and save land for the conservation of biodiversity and natural habitats. Combinatorial biosynthesis is a tool to combine genes from different organisms to produce bioactive compounds in plants. These genetically transformed cultures can produce secondary metabolites in large amounts and can be a promising source for the constant and standardized production of secondary metabolites. Production of potential pharmaceutical compounds using plant transformation strategies and transient expression systems such as agro-infiltration and virus infection is known as molecular farming or biofarming. It focuses mainly on the biosynthesis of proteins and secondary metabolites which are very useful for humans. Plants can act as factories for the biosynthesis of drugs or compounds which are very costly in the markets. There are various bioproducts such as vaccine, antigen, antibody, therapeutical and nutraceutical proteins. Non-pharmaceutical plant-derived proteins are being produced using molecular farming. 4. Summary. In this lecture we learned about in situ and ex situ mode of biodiversity conservation. Biotechnological methods of biodiversity conservation offer many advantages to conventional procedures. Biotechnology benefits agricultural productivity and help in the preservation of biodiversity. Biotechnological methods of preservation have potential for long-term storage. There are two types of cryopreservation protocols that basically differ in their physical mechanisms: classical cryopreservation procedures, in which cooling is performed in the presence of ice, and the procedures based on vitrification, in which cooling normally takes place without ice formation. Classical freezing method includes the successive steps such as pre-growth of samples, cryoprotection, slow cooling 0.52 C per minute to a determined pre-freezing temperature usually around -40 C, rapid immersion of samples in liquid nitrogen LN storage, rapid thawing and recovery. It needs technical skill as it has several steps such as freezing, storage, thawing, reculture of living plant cells during cryopreservation against cryogenic injuries. Maintenance of cryogenic cultures in lane at -196 degrees Celsius or in the vapor phase of lane at -135 degrees Celsius is in such a way that the viability of stored tissues is retained following rewarming. Certain compounds like TIMSO, dimethyl sulfoxide, glycerol, ethylene, propylene, sucrose, mannose, glucose, proline, acetamide etc. are added during the cryopreservation. These are called cryoprotectants and prevent the damage caused to cells by freezing or thawing by reducing the freezing point and supercooling point of water. Various cryopreservation methods are being used for various plant species such as vitrification, encapsulation dehydration and encapsulation vitrification. Cryopreservation using the encapsulation dehydration procedure has been very effective for freezing apices of different plant species from temperate and tropical origin. Vitrification comprises treatment of samples with cryoprotective substances followed by dehydration with highly concentrated plant vitrification solutions (PVS), rapid cooling and rewarming, removal of cryoprotectants and recovery. This procedure has been developed for apices, cell suspensions and somatic embryos of different species. Cryopreservation of seeds is a very valuable strategy for the long-term conservation of tropical and subtropical forest species biodiversity, as it avoids problems related to embryo isolation and in vitro handling. The cryopreserved tissue has considered as safer, clean, disease-free genetic stock. Virus-free strawberry plants could be preserved at 1000 C for about 6 years. Several grape plants have been stored for over 15 years by using a cold storage at temperature around 90 C and transferring them in the fresh medium every year. 3.8 DNA banks. DNA banking is also a potential method for the conservation of biological information by preserving the genomic DNA at low temperatures. The DNA isolation is easy and can be used extensively for the characterization and utilization of biodiversity. The implementation of such biotechnological tools on rare and endangered plant species may help in revival of their previous genes and their products which have been disappeared or inactivated in natural habitat. In gene banks, the plant and animal materials are conserved and are available for breeding, reintroduction, research and other purposes. DNA banks maintain a library of the given DNA sample and provide vital information to the conservation scientists. DNA samples may be of 3 kinds: total genomic DNA, DNA libraries, individual cloned DNA fragments. The maximum possible genetic diversity of the particular genetic stock can be maintained using in vitro gene bank technique. Some important DNA banks includes the Royal Botanic Garden, Kew, UK, presently the world's largest DNA bank, consisting of over 20,000 DNA specimens representative of all plant families. The U.S. Missouri Botanical Garden has collection of more than 20,000 plant tissue samples, and provide raw material for the extraction of DNA for its subsequent use in conservation methods. The International Plant Genetic Resources Institute (IPGRI), as well as the Consultative Group on International Agricultural Research (CGIAR), and International Center for Agricultural Research in the Dry Areas (ICARDA), is heavily involved in the conservation of rare and endangered plant species by maintaining in vitro gene bank. Animal gene banks development is limited by high costs of creation. The recovery and freezing of viable sperm recovered from the epididyms of slaughtered or castrated animals can be a cheap alternative for preserving male gametes. Nowadays, application of post-mortem sperm recovery from the epididymis is available for conservation of endangered species and in constructing semen banks. Preservation of epididymal sperm Preservation of epididymal sperm from accidental dead or slaughtered animals has been reported in several domestic and wild species as boar, cattle, goat, ibex, red deer and sheep. However, epididymal sperm extraction sometimes is not easily practicable in marginal areas, due to the lack of facilities and expertise near the farming areas of local breeds. 3.9 Molecular marker technology. 3.10 Biochemical marker. Different variants of the same enzymes having identical or similar functions are known as isozymes. They are powerful tools for the study of genetic variability within and between populations of plants and animals. They are useful when several taxa, accessions and individuals are to be compared, to identify clones and to examine the clonal structure of various plant populations. They are useful when several taxa, accessions and individuals are to be compared, to identify clones and to examine the clonal structure of various plant populations. 3.11 Phytochemical markers. The discovery of phytochemicals from wild plant species is an achievement toward the eradication of the human diseases. With the advancement of modern techniques such as mass spectrometry (MS) and nuclear magnetic resonance spectrometry (NMR) combined with different separation techniques facilitated the identification and structural elucidation of these phytochemicals. These phytochemical analyses are valuable tools for taxonomic differentiation within species or for evaluating the effect of environmental factors. Variation in biosynthesis of these metabolites could be a result from both genetic and environmental factors, which play important roles in the development of phenotypic variations in plants. 3.12 DNA marker-based techniques. DNA-based markers either PCR-based or non-PCR-based. DNA markers assess genetic diversity based on polymorphism. The polymorphism may be defined as simultaneous occurrence of multiple phenotypic forms of a trait attributable conservation of rare and endangered plant species 3 to the alleles of a single gene or the homologue of a single chromosome within or between populations. Potential DNA-based markers are single-strand conformation polymorphism (SSCP), restriction fragment length polymorphism (RFLP), random amplified polymorphic DNA (RAPD), amplified fragment length polymorphism (AFLP), sequence characterized amplified regions (SCAR), simple sequence repeat (SSR), inter-simple sequence repeat (ISSR), single nucleotide polymorphism (SNP). Those are either PCR-based or non-PCR.